Hello everyone, welcome to this video series specially dedicated to upcoming drug inspector exam in Gujarat state organized by BK Modi Government Pharmacy College. In today's video lecture we are going to discuss about the questions which are asked in previous examination from the microbiology and biotechnology part. So if we see or if we classify the exams broadly which are asked in a previous examination, we can categorize the questions which are asked from the microbiology and biotechnology part into three categories. So the first category is where the questions were asked specifically related to some key characteristic or feature of microorganism. Their special organelles, some anatomical parts, specially they contain. Question were asked from that. Second question or second type of the questions, if we analyze, were asked regarding the specific function of specific part of microorganism, function of function of specific organelles which are present in microorganism and how it is useful to microorganisms from that part. And the third kind of the question, if we see, they were asked from two parts. First is that about that if any specific microorganism they can cause any disease. So the microorganism which cause any disease in a human. And second thing, if microorganisms are used to prepare either vaccine or antibiotic or any other medicine which are used to cure disease. So questions were directly asked regarding that which microorganism are used to prepare what kind of the product. So broadly we can divide the questions into three categories. So first questions or the let us start to discuss some of the questions which are asked in a previous examination. So the first question which we find very often into this question is about the basic building block of the life, that is the DNA and RNA. So as we know, the DNA is the basic building block of the life. So deoxy, it stood for deoxyribose nucleic acid. So as you see in this exam questions, that the questions were directly asked from the basic structure of the DNA. So we all know that structure of the DNA that we right now know, it was proposed by Watson and Crick. So as you can see, the first question was the base pair in a DNA. So the question was about that which bonds are responsible for maintaining the structural integrity of the DNA. And second thing, the questions were from the what kind of the base pair, nitrogen base pair we find in the DNA. So you can see both these questions are related with the basic structure of the DNA. So now if we see the basic structure of the DNA, and because obviously DNA is a very interesting molecule, you can expect lots of questions from that. So let us go through the basic structure of the DNA and then we will shift towards the RNA, because as you know, DNA and RNA are very structurally related at compounds. So let us start with the first DNA. So the DNA, obviously it's a deoxyribose nucleic acid. So if we see the basic structure of basically it's a polymer. You can say it's a polymer because it's composed of lots of monomers. It's composed of the repeated units. As we know it's composed of the repeated units. So what are these repeated units which makes this DNA a very special structure? So if we see the basic structure of the DNA, it is mainly composed of three type of the molecules. So first, it is containing nitrogen base. Second, it is containing a sugar moiety, a 5-carbon sugar moiety. And third, it is containing phosphate, right? Phosphate which is attached to oxygen. So basically, it is composed of three types of the molecule. In a nitrogen base pair also, we know that it is made up of two kind of the nitrogen base pair are there, purine and pyrimidine. So purine and pyrimidine are the two complementary base pair. Now in a DNA structure, because this is the one question is with the correct base pairing, always the nitrogen base pair is a complementary. What is complementary? That means one purine base is always attached with opposite pyrimidine base. So if one is a purine, second is a pyrimidine. If we see the example of the purine, it's a adenine and guanine. Pyrimidine obviously is the opposite one, is the cytosine and thiamine. So one purine base always attach with the pyrimidine base, and this nitrogen base pair is also very specific. Guanine is always attached with the cytosine and adenine is always attached with the thiamine. So what is this, why this is very special is that because this nitrogen base pairing are very specific. Adenine is always with thiamine, guanine is cytosine. What this gives a very specific characteristic to the DNA, and that is that as we know the DNA is a double helix structure, right? Two helix are joined with each other. So because this nitrogen base pairing are complementary, if we know the sequence of nitrogen base pair in a one strand, automatically we know the sequence of nitrogen base in second pair. So to know the entire structure, just information regarding the nitrogen base pair sequence in one pair is enough. So let us see in a figure which will make it more clear. So as you see in this figure, this is the typical structure of the DNA. So as I have explained in previous slide, that basically it is composed of three type of the molecule. So as you see in this one, this is nitrogen base pair, this is 5 carbon ring, that is a ribose, and this is the phosphate. So these three molecules are also attached with three different kind of the bond. As we have explained in the first question, that two complementary nitrogen base pair. So this is one nitrogen base pair, second nitrogen base pair, they are attached with hydrogen bond. But as you see in this structure, this nitrogen base is also attached with this 5 carbon ring structure. So this bond between the nitrogen base and the ribose, 5 carbon ribose, is a glycosidic linkage. It's a glycosidic bond. And the phosphate is attached with this 5 carbon nitrogen base, that is known as a phosphodiesterase bond. So it's a phosphodiesterase bond. So three molecules we have, nitrogen base, then we have this 5 carbon ribose, and we have the phosphate molecule. And we have three kind of the bond: hydrogen bond, glycosidic bond, and the phosphodiesterase bond. Three bonds we are having. Now second key characteristic of this DNA structure is that, as you see, it's a double helix, right? So double helix means two strings, or we can say two strings of the base pair are intervened with each other, or they are making a helix-like structure. And in this one, the direction of this base pairing is also very specific. That means always the base pairs are in opposite direction. So as you see, in this case, if this strand is from 5 prime to 3 prime, so opposite strand is always in a opposite direction. That is 5 prime is over here and 3 prime is over here. And that is why it is known as an anti-parallel, right? So these are the parallel one, but these are going in a different direction. So DNA is a double strand molecule which are having nitrogen base, 5 carbon ribose, and the phosphate molecule, which makes helix-like structure. Generally, the distance between two consecutive nitrogen base pair, it's considered to be around 3.4 angstrom. It is 3.4 angstrom. And so that is, and this complete turn, one complete turn, it's approximately having 10 this unit. It contains 10 units. So one complete turn, as you can see, because the distance is 3.4 angstrom, one complete turn is around 34 angstrom. Right? So this is the DNA structure. So DNA is a complementary base pair. It's a long chain structure where these nitrogens are attached in a sequential mode. So as you see in this DNA structure, the backbone of the structure remains common, that is 5 carbon and this phosphate remains common. The only thing which changes in the DNA structure is the sequence of the nitrogen bond, how they have been attached, how they have been attached with each other. Now, after that, let us see if the DNA structure is clear. Let us see what is the difference between DNA and RNA when it comes to structure part. So, as you can see over here, if we look from the external point of view, the difference between DNA RNA is very common. First difference is that the DNA is, as I said, it's a double helix molecule, right? It's not a single one. It contains 2 strands which are attached with each other. Now compared to that, the RNA is a single strand molecule. So RNA is a single strand molecule compared to DNA. Now the second difference between DNA and RNA, it come to it's about the structure of how they are having. So first is the how they look, single strand, double strand. Now the second critical difference comes between DNA and RNA is in terms of nitrogen base pair. So as we have seen in the DNA, the nitrogen base pair is adenine, guanine, cytosine and the thymine. Now in RNA, these 3 nitrogen base pair, that is adenine, guanine and cytosine, remains common, but they don't contain thymine. It is replaced by uracil. So in RNA, the second difference between DNA and RNA is in terms of the nitrogen base pair. In DNA, we have adenine, guanine, thymine, cytosine. In RNA, you have adenine, guanine, cytosine and uracil. Now the second difference or the third difference between this DNA and RNA comes in the structure of the 5 carbon sugar moiety. So as you see in the DNA, because the word itself is a deoxyribose, so at the second carbon here, it contains hydrogen. But compared to that, in a ribose, it is containing hydroxyl group. So in a DNA, the 5 carbon ring structure is a deoxygenated. And in contrary to that, in a ribose, the second carbon contains hydroxyl group. Now this presence of this hydroxyl group in a RNA make it much more susceptible for hydrolysis process. And compared to that, because in a deoxy there is no reactive group present, it makes DNA a much more stable molecule compared to RNA. And that is why as you see in evolutionary sequence, most of the organisms have shifted for DNA as their genetic material, because DNA is a much more stable molecule compared to RNA. Now this doesn't mean that RNA is not a genetic material at all, right? So DNA in most of the organisms, especially eukaryotic organism, the DNA is the genetic material. But that doesn't mean that RNA is not a genetic material at all. As we will discuss in our subsequent lecture, that there are many viruses which contains RNA as their genetic material. So that also comes to a second explanation regarding difference between DNA and RNA. As we know that to be act as a genetic material, because basically genetic material is required to pass information from generation to generation, the molecules required to have a very critical property, and that critical property is something known as a self-replication, right? So what is self-replication? That means that we know that the DNA molecule can replicate itself. That means a DNA molecule can generate the same copy on its own. So that is self-replication molecule. Generally, in a eukaryotic cell, RNA cannot replicate themselves. But when RNA acts as a genetic material in some of the viruses, these viruses contain some enzyme, they contain mechanism by which they can also replicate the RNA molecule. So it's not that self-replicating is a very, what we can say, unique feature to DNA only. RNA can also replicate itself, but mostly in a eukaryotic cell they don't do. Whenever RNA acts as a genetic material, they also have this ability to replicate themselves on their own. So let us summarize the difference between DNA and RNA, because I think this structure of the DNA and RNA, as you see, there are lots of critical part of this structure, and that is why you can expect questions from the structure of the DNA and RNA. So as I have summarized the structure between DNA and RNA over here, so you can see that it comes in a mainly three part. First is the how it looks. So double strand, single strand. Then is the presence of hydroxyl group in case of the RNA, and it's a deoxy. That means it contains only hydrogen in case of the in case of the DNA. Second, it comes a nitrogen base pairing. It contain uracil. It contain thiamine. So these questions, if the question are asked from the DNA and RNA, so this basic structure of the DNA and RNA is like that. So let us see the next question. Okay, second type of the question which I have find in the previous exams were related with something called like this, the father of microbiology. So now what these questions are there? There are many great scientists who have contributed immensely in the field of the microbiology. So these kind of the questions, obviously because our basic field is related with the medicine or microbiology, or we can say living organism, the scientists which have contributed in that area are important. Obviously it is important from the perspective of the pharmaceutical syllabus, and also if we see the one portion where we are going to deal with the GK question, in that also we can find questions where they have asked about the contribution of the specific scientists. So the history of the microbiology, obviously the question was where it was a very simple question. The father of the microbiology, Anthony Leeuwenhoek, is the answer they have suggested. But by going through answer, I thought that let us discuss, or let us go through the famous scientists which have contributed immensely in the field of the microbiology or related with the pharmacy. So these questions can be useful in a two part. It can be general GK question, and also this question can also be asked in a pharmaceutical part. So let us see, I have pointed out some of the few scientists which innovation has been, whose innovation considered as a breakthrough, or what we can say, it's a breakthrough innovation in the pharmaceutical industry. So as you see, I have listed some of the great scientists. Now in this one, the question was basically, question is who is the considered as a father of the microbiology. So generally there is no all agreed answer on that, because based on the nation, they consider different person as a father of microbiology. So obviously Anthony Leeuwenhoek, he is considered as one of the very great contributor in the field of the microbiology. He was the person who observed the first living cell, and he also contributed in some other theories, so he also considered. But if you see some of the books or some of the articles on the internet, where they have also mentioned the name of the Louis Pasteur, right? As he's a very famous scientist from the France, and we all know he has also been named or given the credit to be considered as a father of the microbiology. So in this question also, you see the name of the Louis Pasteur. So these questions can be, there is no specific answer what I can say, because all these scientists have contributed. But let us not consider what's the father of microbiology. Let us consider what is the contribution these great scientists have made into the field of the pharma, and that is why they are regarded as a why they are been so respected in the field. So obviously Louis Pasteur has worked in many fields, and he has contributed in lots of areas, especially when it comes to pharmaceutics. So obviously Louis Pasteur, he has innovated this pasteurization method, still we use for the milk. Then obviously he was also front runner in the vaccine development also. So he has also tried to develop the vaccine for rabies, cholera. He worked on that. So he also worked in lots of areas. So one thing is that the father of the microbiology, the opinion can be varied. But when we say father of the bacteriology, Louis Pasteur is all accepted one. So when we say Louis Pasteur, father of the bacteriology, because he works on basic bacteria and their theories, so he's respected as a father of bacteriology. Then obviously another one, that is Alexander Fleming, who invented the penicillin. As we know, penicillin was the first antibiotic that was innovated, and after that we entered into the area of this antibiotic. So that is the one of the great contributor. Then because nowadays vaccine is in spotlight, so obviously the father of immunology or the father of vaccination is the Edward Jenner. As we know, he has innovated the smallpox vaccine from the cowpox, and he tried on the patients, and then he discovered the cure of the vaccine. So when it comes to vaccine, the Dr. Edward Jenner is considered as a father of vaccinology. Now, third one, because why I have chosen, why I was selected this another scientist is that because as we know right now, the war is going on in Russia and Ukraine, and one of the scarcity it has produced in the world is regarding the supply of the wheat, because wheat is one of the very essential food ingredient throughout the world. So because right now the shortage is going on, everyone is talking about the green revolution, and because India has one of the highest stock of the wheat we are having, and everyone is asking India to supply, but that was not the case in India, right? If we see in 1965 to 70, we were living on imported wheat, and that has changed drastically once the green revolution has happened. So the green revolution has really put the world on a different perspective, and that is why two persons who have contributed greatly to this green revolution, you can say, which has enabled farmers to produce lots more food, wheat, rice, and because of that the countries like India and other countries which have become self-sufficient in terms of the food production. That was not case in 1970. As I said, we were on imported grain, but right now India is one of the topmost exporter in the wheat. So scientist Dr. Norman Borlaug, he is considered as a father of the green revolution. He started to experiment with this high quality, high producing wheat crop, high yield producing wheats, and they have started the experiment with them, and and that is why he is considered the father of the green revolution in the world. But when we say in India, father of the green revolution is considered Dr. M.S. Swaminathan. So M.S. Swaminathan, he started to use this technology, and they have come up with this plant crops which were giving high yield per acre into India, and then farmers in India started to use this crop variety, and because of that efforts, right now India at a position where we become net exporter rather than the net importer. So M.S. Swaminathan has contributed greatly, and that is why he is considered as a father of the green revolution in India. So if you remember, when in 2014 when our Prime Minister went to US first time, and he was giving speech in New York. In that speech, he specifically mentioned that India is thankful to Rockefeller Institute and Dr. Norman Borlaug because they developed this high yield crop variety, and because India used that crop variety, and because of that we were able to become self-sufficient in our food. He specifically thanks all these scientists who have contributed into this one. So that is why Dr. Norman Borlaug and M.S. Swaminathan, they are father of the green revolution. Then Robert Koch. So Robert Koch was also the doctor. Basically, he come up with the germ theory of the disease. So what he observed was that there are many diseases in a human that are due to the infection of the microorganism. So he was the first one who started to analyze the blood for appearance of the microorganism, and then he stated that there are many diseases in the human which are due to the infection of the microorganism. That is known as a germ theory, and that is why due to that innovation he is considered as the father of the medicinal bacteriology, because he was the first to propose this theory that infection from microorganism can result in a disease, and he started to observe the blood for the infection. So that is the Robert Koch. Then another important discovery was the streptomycin. So streptomycin was I think it was another very important antibiotics discovered. So streptomycin, it was discovered by Salman Waxman and Albert Setz. So there is a controversy between that who discovered that one. Basically Salman Waxman was a PhD guy and Albert Setz was a student. So they discovered the streptomycin. It's also one of the very effective antibiotic that was discovered. And second last one which I think is that is the Human Genome Project, because Human Genome Project has been always been in a discussion. The fruits of Human Genome Project are also right now scientists are using to prepare to find out new targets for the drug discovery, to discover the new kind of the drug. These results of the Human Genome Projects are still very much in discussion. So the Dr. Eric Lander, who is a professor at MIT, Massachusetts Institute of Technology, basically it was his brainchild. He conceived this project and then executed with various universities by taking in them. So this is not the exhaustive list. As you see, I have only mentioned few of the one which I think that are useful not only in the pharmaceutical field or related to medicinal field, but this question can also be asked in a general GK question. So that is why these innovators and innovations are important. So let us see next one. Okay, so the next questions which I find, lots of questions which I find is based on the basic difference between prokaryotic and the eukaryotic cells. So as you see this very simple question, which of the following doesn't contain mitochondria? So the answer was bacteria. So basically this question comes under the banner of difference between prokaryotics and the eukaryotics. So what is basic difference between, let us discuss the basic difference. So prokaryotes and the eukaryotes, the word itself starts from the karyotes. So karyotes is a nucleus, right? So prokaryotes means, pro means before. So when the initial organism started to appear on earth, they are not having this kind of the distinct nucleus which can contain the genetic material. And that is why they are known as a prokaryotes, because they are not having this kind of distinct nucleuses. Now U stood with and karyotes mean the nucleus. So the organisms which are containing specific nucleus where our genetic material resides or they confined inside, they are known as a eukaryotic organisms. So prokaryotics are, you can say, they are the basic organisms which are not containing this kind of specific genetic material. Then the question is that if they are not having this kind of specific nucleus, where their genetic material rise? So their genetic material generally floats in a cytoplasm. So it's not confined to anyone, any boundary. They just float inside the cytoplasm. So what are the basic difference between prokaryotes and the eukaryotes? Let us see what is the basic difference. Okay, so as you see here, this is the typical structure of the prokaryotic cell. So the ubiquitous examples of the prokaryotic cell is nothing but it's a bacteria. So bacteria is a example of the prokaryotic cell. And eukaryotic cell, you can take any cell, human cell, plant cell, animal cell, these all are eukaryotic cell. So if we see the basic difference between prokaryotic cell and eukaryotic cell, it can basically it can categorize into three categories. So the first category comes, as we have discussed, is it at the place something called nucleus, right? So if you see in the eukaryotic cell, it is containing a specific nucleus which confines genetic material. So it's a nucleus which contains chromosome, right? Inside we said chromosome. And as you can see in the prokaryotic cell, there is no this kind of the structure available which can protect or which is surround this genetic material. And as you can see in the prokaryotic cell, genetic materials are floating within the cytoplasm. Now second difference comes in a eukaryotic cell is that in a eukaryotic cell, the chromosomes are generally linear, something like this, as we see this one. In case of the prokaryotic cell, it is circular. It is something like that. Right? So it's a circular one. It's a linear one. Another thing difference between comes in the eukaryotic cell and prokaryotic cell is that in a eukaryotic cell, the DNA is wrapped around a protein called histone protein. So it is around histone protein. Now the reason is that as we know the eukaryotic cells, the DNA of the eukaryotic cell is a very lengthy one, right? It's contain billion base pair. So to make sure that this length, or we can say all this lengthy DNA can confine within this tiny nucleus, it is wrapped around a special protein called histone protein. So histone proteins are specific protein around which our DNA gets wrapped, and which is a very unique feature of the eukaryotic cell. In a prokaryotic cell, no such a histone proteins are observed. It contains only DNA. There are no histone proteins are available. Now, third difference between comes in the prokaryotic and eukaryotic cell is that there are specialized organelles which are only specifically found in a eukaryotic cell. For example, mitochondria, Golgi apparatus, then lysozyme, right? So these are the specific organelles which are only specifically present into the eukaryotic cell, and also something called endoplasmic reticulum where the ribosomes rRNA resides. So all these specific organelles, right? Mitochondria, I don't require to explain its function. We know it's a powerhouse which generates lots of energy, which generates ATP, helps in generating ATP. Golgi apparatus, lysosomes, endoplasmic reticulum, these are specifically found only eukaryotic cell only. They are not found in the prokaryotic cell. So that is the second difference. Now the third difference come is that all the prokaryotic cell are having very good developed cell wall. So you can see the cell walls are developed cell wall present in a prokaryotic cell. In a eukaryotic cell, this cell wall is not it's not an essential integral part. Some eukaryotic cell contains cell wall, some doesn't contain cell wall. For example, plant cell are having good cell wall, right? But animal cells they don't have a cell wall. But in a prokaryotic cell, cell wall is there, but because it is one of the essential part of their protection mechanism, and that is why they contain a very good cell wall. Second thing, the difference between prokaryotic and eukaryotic cell is that generally in a prokaryotic cell they contain only single working chromosome. So it contains only single chromosome, where in case of the eukaryotic cell we know that our chromosome is in a pair, right? So we have the pair of the chromosome. In case of the prokaryotic cell is the single chromosome. And second thing, in a prokaryotic cell it is not essential, but sometimes it is observed in a prokaryotic cell is something called plasmid. What is plasmid? Plasmid is an extra chromosomal DNA. It's an extra chromosomal DNA. It's a DNA only, but it is extra chromosomal. That means it is not going to produce, it doesn't produce generally protein which is required for the regular functioning. So plasmids are DNA which is present outside of the, it is present besides the main chromosome in a prokaryotic cell. It's an extra chromosomal DNA, and this DNA can also have a self-replication ability. They also replicate on their own. So these plasmids are not present in a eukaryotic cell. In a eukaryotic cell, the DNA which is required or the in form of the chromosome is present inside the nucleus only. So to answer our main question which was asked in So as you see, the question was that which of the following doesn't contain mitochondria. So mitochondria, bacteria don't have the mitochondria. Eukaryotic cells have the mitochondria, because mitochondria is a function of its mitochondria is a function of producing ATP. They produce more ATP, right? It helps in a metabolism. Okay, so you can see the difference I have summarized over here between the prokaryotes and eukaryotes. So you can see both have the, what are the common characteristic between? So both have the DNA, obviously have a DNA, but in a eukaryotic cell, DNA is a linear, here is the circular one. Both have the cytoplasm, both have a cell membrane, both have the ribosome. But what are the unique feature of the prokaryotic cell? Obviously, no nucleus. Small and simple, no organelles. Organelles means they don't have the mitochondria, they don't have the Golgi apparatus. That is not like that. Mostly are unicellular. The prokaryotes are the unicellular in nature, and examples are the bacteria. So the second question which you can get from this discussion is that plasmid is obtained from. So obviously plasmid is a characteristic of the prokaryotic cell, and prokaryotic cells are example of the bacteria are the example of the prokaryotic cell. So you can answer this one, that plasmid is generally obtained from the bacteria. Now if we are since we are discussing the bacteria, in a bacteria also one key component, or we can say the key part of the bacteria which gets lots of question or discussion is about the cell wall. As I have said that prokaryotic cells are characterized by well developed defined cell wall. So that is why lots of questions you can see from the cell wall part. Now generally in our pharmaceutical microbiology, we all know that bacteria can be classified into gram positive bacteria and gram negative bacteria based on the composition of the cell wall, and that is what you can see these two questions come from. So bacterial cells are more resistant to osmotic shock than eukaryotic cell, because why? So because it is containing its cell wall is made up of the peptidoglycan, and second is about the something about the endotoxin present in a gram negative bacteria. So let us see that what are the key characteristic of the bacteria. So if we see the structure of the bacteria from the outside, mainly some bacteria, as we know, they contain a very good capsule. So capsule is a layer which is around the bacteria. Generally it is due to the metabolic end product started to deposit around the bacteria. So some bacteria are containing capsule, some are not containing capsule. Cell wall is an integral part. All the bacteria contain cell wall. Then cytoplasm, plasma membrane, then cytoplasm, plasmid, ribosome, everything is inside the cytoplasm. So in that, as I have said, the key or one of the identifying feature of the bacteria is the cell wall. And as we know that based on the cell wall composition, we can classify the bacteria in the two category, that is known as a gram positive and gram negative bacteria, right? So this gram word comes from the method which was innovated by Dr. Gram. He innovated this method to differentiate the bacteria based on their cell composition. So let us see what is the cell, how unique is the composition of the cell wall of the bacteria, and how based on that we can classify the bacteria, or how we can differentiate the bacteria based on the cell wall composition. So as you see over here, the bacteria can be classified into two categories based on what kind of the cell wall they are containing. So you can see gram positive, gram negative bacteria. So what is the key characteristic of the gram positive bacteria is that the gram positive bacteria contain a very thick layer of peptidoglycan. So this is containing a very thick layer of peptidoglycan layer. So peptidoglycan, it's peptide plus glycan. So you can see amino acid and sugar. In simple words, we can say it's a peptidoglycan layer. It contains peptidoglycan. So very thick layer of peptidoglycan. And along with that, it also contains lipoteichoic acid and teichoic acid into that composition. But majority of the cell wall is composed of peptidoglycan. Now, contrary to that, if we see the structure of the gram negative bacteria, it is not that they don't contain peptidoglycan at all. They contain peptidoglycan layer, but this peptidoglycan is a very thin layer. So you can see, compared to this gram-positive bacterial peptidoglycan layer here, the peptidoglycan layer is very thin layer. What is the key characteristic of gram-negative bacteria is something called this structure that is known as a outer membrane. So this outer membrane, which is largely composed of lots of lipid, are key characteristic of the gram-negative bacteria. So if we see the structure of cell wall for gram-negative bacteria, it contains a thin layer of peptidoglycan, a layer of lipoprotein, and a large layer of lipopolysaccharide. So you can see it contains lots of lipid in the structure. So one of the major difference between gram-positive bacteria and gram-negative bacteria is that in a gram-negative bacterial cell wall, the lipid content is very high. And that is why you see the drug which we use to treat gram-positive bacteria and gram-negative bacteria are different. Why? The reason is that the drug required to pass through different pathway. In a gram-negative positive bacteria, it has to pass through peptidoglycan layer. And in a to enter in a gram-negative bacteria, they are required to pass through this lipidic layer, and that is why the drug which are effective on gram-positive and gram-negative are different. So in a gram-negative bacteria, the key composition is that it contains large amount of the lipid in form of the polysaccharide or in form of the lipoprotein, but it contains lipid, and it also contains something called porins protein, which is a facilitation way, or which is the way for transport of the molecules. It can be water molecule, or it can be amino acid, or whatever essential ingredient that required. Porins protein give a passage for the transport, right? So basically lipopolysaccharide and the lipoprotein are the key characteristic of the gram-negative bacteria. So and we also know that based on the structure of this gram-negative and gram-positive bacteria, we can differentiate by using gram staining technique, right? So gram staining technique is used to differentiate the bacteria based on the structure of the cell wall. So gram staining is one of the important technique which is used to differentiate the gram-positive and gram-negative bacteria, and that is why you see these two questions which are directly asked from the gram staining technique. So question was that what is the purpose of safranin in a gram staining technique, and second thing is that which of the following organism has a teichoic acid as a part of their cell wall. So as we know, teichoic acid and glycoteichoic acid is a part of the gram-positive bacteria, as we have seen in the figure, right? So teichoic acid and lipoteichoic acid are the part of gram-positive bacterial cell wall. And also the question was from the gram staining. So let us see what are the steps in a gram staining. So I think we all have performed this practical, but still let us make the principle only clear. So gram staining basically a technique where we differentiate the bacteria based on their cell wall composition. The basic differentiating feature between gram-positive and gram-negative bacteria is that it's a lipid content. So as we know, in a negative there are lots of lipid. So what are the steps in a gram staining technique? So the first we make a smear. Once the smear is prepared, then it is stained primarily with the crystal violet. So crystal violet is a primary stain. So once we use this crystal violet, all the bacteria, the cell wall of the bacteria becomes blue because crystal violet is a blue color. Then we add iodine. So iodine makes the complex with the crystal violet, and this complex with the crystal violet makes sure that this primary stain it joins very strongly with the cell wall containing peptidoglycan layer. So once we add iodine, it makes a strong complex with the crystal violet, and which makes sure that the strong bonding is formed between the primary stain and the cell wall. Now what happens when you use alcohol and acetone in our third step, which is also known as a decoloring or differentiating agent, because the gram-negative bacteria contain lots of lipid, and we know that lipid is soluble in organic solvent, alcohol or acetone. When they comes in contact with the alcohol or acetone, this gram-negative cell wall leaches the color. Whatever color they have obtained during the treatment of iodine and violet, once they come in contact with the alcohol, they leaches the color. So because the lipids are soluble in alcohol, it will allow this color to move away. And because of that, once we treat the gram positive bacteria, gram negative the smear with the alcohol, the gram negative bacteria will lose the color. And once the decolorization step is over, the gram positive bacteria remains blue color. Why? Because they are not containing lipid, and because they don't contain lipid, they will not leach the color. Contrary to that, the gram negative bacteria which contain lipid, they become colorless. And once the decolorization over, then we use safranin as a counter stain. So to differentiate the blue bacteria, the gram positive bacteria which is of the blue color from the gram negative bacteria, we add safranin. So now safranin only stains the gram negative bacteria and it becomes pink. So when we observe the slide, the gram positive bacteria will appear as a blue color and the gram negative bacteria will appear as a pink color. So safranin is a counter stain, the moderant is the iodine and the crystal violet. So basically we require 4 components for the gram staining: crystal violet, primary stain, iodine which is a fixator or the mordant, decolorizing agent or differentiating agent, alcohol or acetone, and counter stain is the safranin. So you can see this color. What is the question? What is the purpose of safranin in gram staining? It's a counter stain which stains the gram negative bacteria with the pink color. So as we are discussing about the bacteria, there is a special class of the bacteria which holds a special consideration. So as we have discussed lots of about structure of the bacteria, mostly right up to now we have discussed about the various part of the bacteria, especially the cell wall, cytoplasm, DNA. Now one of the important characteristic of the bacteria is that the prokaryotic cell or the bacteria, they contain a specific enzyme, something called endonuclease. So what is endonuclease and how it works? So so bacteria, what happens with the for the bacteria, viruses are villain. So what is viruses and how they work? Basically we know what is viruses. Viruses are a link between living and non-living, right? So when viruses are outside of the suitable host, they don't do any metabolic activity, and viruses are in always constant search of the suitable host. So what happens is that when viruses, when they are outside, they are not doing any metabolic activity, right? But when they comes in a contact with the suitable host, what viruses do? We all know virus get attached to this host and they will insert their genetic material inside. Now once the virus insert the genetic material, we will discuss in our subsequent lecture when we discuss specifically virus. What they do is that they take all the machinery, all the nutrition that this cell is having and utilize this machinery to replicate its own genome. And because the viruses use all this machinery of cell and make lots of copies, after some times what happens that this cell get exhaust of all the nutrition. And once the cell is exhausted of all the nutrition, obviously you can see the cell will die. So that is why what I read is that for bacteria, viruses are villain. So what happens is that viruses attacks the bacteria, they utilizes this machinery and ultimately kills the bacteria. So that is the regular what we can say cycle virus is having. So bacteria also had devised some mechanism by which they can protect themselves from this virus, right? Because virus is acting as a villain for the bacteria, and bacteria has also devised some mechanism to make sure that these viruses, whenever they are getting inside the bacteria, they cannot utilize the machinery of the bacteria and they cannot replicate themselves. And that is why bacteria has devised a mechanism, or they produces the enzyme, something called restriction endonuclease enzyme. So restriction endonuclease enzyme is a specific enzyme which bind with the specific sequence. They will identify the foreign DNA which is entered into the cell, and when they identify it, they will cleave it, they will destroy it. So what happens, that whenever virus enters into the bacteria, right, this restriction endonuclease enzyme, they bind the specific sequence, they identify the specific sequence, bind with it and destroy it, so that this viral DNA cannot hijack the machinery of the bacterial cell and replicate itself. So basically restriction endonuclease is a protective mechanism that bacteria is having, which protects the bacteria from cleaving or destroying the foreign DNA. So, and what is the key characteristic of this endonuclease? Because remember, bacteria also having its own DNA. So this restriction endonuclease should not attack its own DNA. And that is why restriction endonuclease doesn't work randomly. It only identifies a specific sequence first, bind with that specific sequence, and then only it start cleaving. So it's a selective one. So what is the restriction endonuclease enzyme? Basically restriction endonuclease enzyme is the defense mechanism which is present in the bacteria, which protect it from the foreign DNA. You can say it's a virus, basically it protect from the foreign DNA. So you can see the question was that which enzyme cleaves the DNA. So enzyme which cleaves the DNA is something known as a endonuclease. So endonuclease, restriction endonuclease, which presents into the bacteria, it protects it from the viruses or the foreign DNA. Now on the same way, the second question which was also there, which I found in the paper, is that which of the following enzyme act on the DNA after its entry into the cell. Now this question was not controversial, but it can have a two answer, right? Basically nuclease enzymes are the enzyme which cleaves the DNA, but you can see in this question the option was also given endonuclease. So endonuclease also cleaves the DNA once it enter into the DNA. The answer which was suggested by the GPSC was deoxyribose nuclease. Now I am not saying deoxyribose nuclease don't cleave. It also cleave the DNA, but so does also the endonuclease. So this question, right, it's not something you can go specific answer for. Generally all the nuclease enzyme has this ability to cleave, but restriction endonuclease, because it resides inside the DNA, bacteria, and they cleave the foreign DNA, it also cleaves only once it get enter into the one. So here according to me, right, the endonuclease is also not a wrong answer, but the deoxyribose was suggested, deoxyribonuclease was suggested by the one. So it's also the ribonuclease, it is also having the same function. But what is the difference between deoxyribose and the endonuclease is that endonuclease required a specific sequence. It only work when they find out the specific sequence. It cannot work randomly. So endonuclease required a specific sequence to cleave. That is not the case with the deoxyribonuclease. Deoxyribonuclease also cleave the DNA, but endonuclease is a specific enzyme for the protection of the bacteria so that the viral DNA cannot infect it. And second thing, it is also DNA specific one. That means a specific DNA is identified and then it cleave it. So third question was also there. You can see this one. Which of the following enzyme in bacteria is responsible for restricting the growth of the virus? So obviously is the restriction endonuclease enzyme which is responsible for restricting the growth. So here this question is slightly what we can say controversial, but according to the literature, restriction endonuclease enzyme, it's one of what we can say main enzyme which protects the bacteria from viruses. So you can see over here how this restriction endonuclease work. So basically when this virus is enters into the bacteria, this restriction endonuclease enzyme, it cleaves it, it degrades, it cuts this DNA sequence and protect the bacterial chromosome. So that is the main restriction endonuclease enzyme, how it works. So another question which is restricted one. So depending on from where this restriction enzyme has been isolated, there are lots of restriction endonuclease enzyme has been identified, and they are given the name. For example, E. coli R1, which is isolated from E. coli, Hind, then the one. So depending on BAMH1, so there are lots of restriction endonuclease enzyme right now in a application. So as one of the main application of this restriction endonuclease enzyme when it comes to biotechnology is that these enzymes, because they can identify the specific sequence, they find their applicability in the genetic engineering. When we require to cut a specific sequence of the gene, and then this gene is joined with the vector to be introduced into the host or the host cell. So this restriction endonuclease enzyme, be it a natural protector for the bacteria, but it finds a very immense application when it comes to genetic engineering. So you can say that the question was that which is not a restriction endonuclease enzyme, and DNA ligase was the option, because you know DNA ligase is an enzyme which we see in the DNA replication process. So when we discuss the DNA replication process, we know the DNA ligase enzyme is responsible for joining the two DNA sequence rather than the cutting the DNA sequence. So this question was also, you can see, from the restriction endonuclease enzyme. Now in this bacteria, one bacteria which find a specific mention in this whole range of the bacteria is the acid fast bacteria. So acid fast bacteria, Mycobacterium tuberculosis, as we know, it is main is a Mycobacterium tuberculosis and Mycobacterium leprae, right? So two bacteria which find special mention. The reason is that both cause a very grave disease. Mycobacterium tuberculosis responsible for TB, and Mycobacterium leprae, which is responsible for leprosy. So both these bacteria. Now why this bacteria required specific mention is that because as we discussed in our gram positive gram negative bacteria, that the key characteristic of the gram negative bacteria is containing lots of lipid, and that is why the drug which act on a gram negative bacteria are different from the gram negative bacteria. Now here in a mycobacteria, the key characteristic of the mycobacteria is that they contain a very thick layer of mycolic acid. They contain a very thick layer of mycolic acid. And because it contains very thick layer of mycolic acid, the drugs which generally act on a gram positive bacteria are not applicable on this Mycobacterium tuberculosis, Mycobacterium tuberculosis bacteria, right? So mycobacteria, if we see, if you remember the key part of a gram negative bacteria, as we discussed, in a gram negative bacteria, the key characteristic of the gram negative bacteria was the outer layer. It was containing a very good outer layer, well developed outer layer, which contained lipopolysaccharide, then lipoprotein, and like that. And that is why the drug we required to pass all this lipid layer to enter in a gram negative bacteria. But in a mycobacteria, they are not containing this outer layer, but contrary to that, they contain a very thick layer of the mycolic acid. It's a thick wax-like layer of mycolic acid, and that is why generally the drugs which affect you on a gram positive bacteria doesn't affect the Mycobacterium tuberculosis because of the thick wax layer. And that is why, because of this thick layer, if we try to stain this Mycobacterium tuberculosis with our gram staining technique, typical gram staining technique, it will not get stained. Generally, mycobacteria is a gram positive bacteria. They are classified as a gram positive one. Why? Because they don't contain outer layer, and because they don't contain outer layer, they are classified as a gram positive. But because they contain a very thick layer of mycolic acid, they will not stain with the regular gram staining technique, and that is why we required a specific staining technique to stain the Mycobacterium tuberculosis bacteria. Right, and that is even though they are gram positive, they require a specific technique where we can penetrate this thick layer of mycolic acid, and this technique is known as a Zell-Nelson method. So you can see the Zell-Nelson method, how it differentiates from the regular. Okay, so in a Zell-Nelson method, the primary stain is a carbon fused chain. Primary is a carbon fused chain. So carbon fused chain is a primary stain along with the phenol. So carbon fused chain plus phenol are added to this primary, uh, to this smear as a primary stain. Now, as I have said that because this contain a very thick layer of mycolic acid, this carbon fuchsin and phenol will not able to penetrate inside, and that is why we require to also apply heat so that the cell wall becomes somewhat porous and the primary stain can penetrate inside. So the carbon fuchsin plus phenol is a primary stain. Along with that, we also required to apply heat. Once we applied the heat, then this cell wall becomes somewhat porous and our primary stain will enter. Once this primary stain is enter, so that all the mycolic acid containing bacteria, that is our acid fast bacteria, will stain, right? So now what we required in a second step, the other bacteria which are not containing mycolic acid, they should lose the color. So in a next step, that is known as a decoloration step, generally acid plus alcohol. So combination of acid and alcohol is used as a decoloring agent. In a gram staining, it was only acetone or alcohol was enough. But here acid and alcohol are used, and once all the known acid fast bacteria loses the color, they are counter stained by methylene blue or crystal violet. So because these mycolic acid are hard to penetrate, we required phenol and all this heat to make sure that this primary stain enters into the bacteria. And once they are primary stain, then other bacteria are decolorized by using acid alcohol and counter stained by the methylene blue. So Mycobacterium tuberculosis, they required a special mention in the category of the bacteria because of two reasons. First, it causes a disease, tuberculosis, and second thing is that it's the typical cell wall structure. Even though they are gram positive, even though they don't contain outer structure or outer cell wall like in the case of gram negative bacteria, but they are not easily stained in a gram staining technique. They contain a thick layer of the mycolic acid, which makes them special for in terms of the staining and also in terms of the effectiveness of the drug. So you can see this example. So which of the following bacteria is acid fast microbes? Obviously Mycobacterium tuberculosis and Microbacteria leprae, two are the acid fast one. And which bacteria grow in acidic pH? Lactobacillus. Lactobacillus, we know it's a part of our day-to-day food, right? Lactobacillus. So lactobacillus, we all know they grow in a acidic. They grow in acidic one. So this question specifically related with the acid fast staining, it's very important one because these bacteria are having the specific characteristic which are explored in this Zell-Nielsen technique. So let us stop over here. Thank you everyone for the patient listening. Please share your comments, share your experience on our channel, and try to help us be better. Thank you everyone.